Okay, can you hear me? Come on, let's begin the story. Good morning, all of you. Hi. Okay, done. Up to the last class, I was discussing about xylem, complex tissues, xylem. Let us begin the another part of the complex tissue, phloem. Phloem. Important these parts. From here you maybe get some confusion. Okay, carefully follow me. We will go without any confusion. Okay, see, phloem. Phloem is nothing but the food conducting tissue. In other words, it is also called bast. Bast. See, the bast major work of the phloem is transport of food material. Food means what sort of food it will actually transfer or transport. It transport food in the form of sucrose. The only one form of food phloem can transport, that is sucrose. Only one form, sucrose. Why? Later we will see. Next one. The mode of transportation is always multi-directional, which means, as we have seen in earlier, root to leaf, plant can transfer water unidirectionally through xylem. But when it comes to phloem transportation, the transportation is multi-directional. It can take food from leaf to root, root to branches, branches to again root. Any direction food can able to be transported through phloem. That's why phloem transportation is multi-directional. Xylem transportation is unidirectional. Then let us talk about in angiosperms. What are the tissues present in phloem? Phloem made up of sieve tubes, companion cells, BOTANY cells, companion cells, phloem parenchyma, and also phloem fibers. These are the components present in phloem. 4, not a big deal. But before going to their description, let us check in gymnosperms. What is the condition? Is it okay? We will talk about all those things. Somebody saying protoxylem, metaxylem. Don't worry. Step by step. Clarity. Okay? We go in a sequence. Then only you can remember better. Suddenly, one stone, if you throw, it may hit or not. But observe, if you are practicing throwing stone consistently, definitely you can have the chance to hit. So let us move step by step slowly. Okay, let us observe. In case of gymnosperms, what is the composition? They are also vascular plants. What are the kind of tissues they possess? They possess sieve cells. They don't have sieve tubes, keep it in mind. And they do not have companion cells. Instead of companion cells, they have albuminous cells. The replacement of companion cells is albuminous cell. Next one, they have parenchyma as usual, and they even possess fibers. Mostly phloem fibers will be there, but xylem fiber in gymnosperm is a questionable thing. Most of the gymnosperms do not have fibers in their xylem, but phloem fibers will be there in most of the gymnosperm. Okay, let us see one by one about what I am going to discuss. Look at this. I will not talk hereafter anything associated to gymnosperm. That's not our discussion. Our discussion is anatomy of flowering plants, nothing but angiosperms. So let us stick on to angiosperm, phloem structure and their detail. In that, fibers are dead. Fibers are dead. The one and only dead tissue that could be noticed in phloem is fibers. In earlier class, when I was talking about sclerenchymatous fibers, I have given one hint to you. Wherever you come across a word fiber, that is nothing but sclerenchymatous fiber. In the plant body, other variety of fibers are not available. There is no parenchymatous fiber. There is no collenchymatous fiber. No. There is a fiber that is nothing but parenchymatous fiber. Clear? Then, sorry, sclerenchymatous fibers. Next one. Sieve tubes, companion cells, parenchyma. All are living cells. They are all are living cells. Living. What do you mean by living? They have protoplasm, whether they have nucleus or not. They have protoplasm. Their walls are thin, made up of parenchymatous cells. Then, if you are talking about with this criteria, phloem is known as living connecting tissue or living conducting tissue. Conducting means transportation. Phloem is living conducting tissue. In that account, when we were talking about xylem, we mentioned xylem is dead conducting tissue. Because of its dead nature, it even can provide mechanical stability. Wherever you come across phloem has one and one role. It performs transportation of food and water, never ever provide mechanical stability. Mechanical stability is not an account of phloem. Okay? Then let us see one by one how these cells will arrange and their nature. I am talking hereafter only associating information of angiosperms. Our discussion is not about gymnosperms at all. Then sieve tubes. Sieve tubes. If you talk about sieve tubes, it is not single cell. Several cells called sieve tube elements. Sieve tube elements fuse and form sieve tubes. Sieve tube is not a single cell. I think you remember the same word even I have given to you when we were talking about vessel. Vessel is not a single cell. There are vessel members, they fuse together. Ultimately, there is a formation of vessel. Vessel made up of vessel members. Similarly, sieve tube made up of sieve tube elements. Let us check how these sieve tube elements will form sieve tube. Consider this. This is a sieve. This is a sieve tube element. I am saying this is a sieve tube element. Sieve tube elements will have obliquely arranged cross walls. This is one sieve tube element. Generally, this is general appearance. Okay, I am saying this is a sieve tube element. Sieve tube element, which means one cell. This cell, you can see, one will be fused with another. This overall body is known as sieve tube. One is called sieve tube element. So, characters. They have cell wall, but the cell wall is primary. You know what does it mean by Primary wall. Primary wall made up of cellulose. Yes or no? Cellulose. Okay. Next, one sieve tube element wall will fuse with another sieve tube element wall. Then there will be a formation of a kind of connecting wall between the two sieve tube elements. That connecting wall is known as perforated end plate. This perforated end plate is appeared like a sieve plate. Sieve plate is nothing but perforated end plate. Perforated end plate. The word perforated end plate, even you have seen between the two vessels. Same, but their secondary walls, here primary walls are having certain amount of pores. This is sieve plate. Then go to one cell. Inside one cell. Inside one cell, you will see a large vacuole. Large vacuole. This vacuole has been formed in order to make the cell to be elongated. We want to make the cell to be enlarged, not in size wise, in the length wise. So while we are enlarging the cell, there is a development of something that is called vacuole. Vacuole develop. Around the vacuole, there is a presence of peripheral cytoplasm. Cytoplasm is peripheral. Then if you see inside the cell, they are anucleated, or some another word is there, anucleated or enucleated. Both are same but different. What is that? Imagine if a cell has no nucleus, that cell I will call it as anucleated simply. It has no nucleus, that's why I am saying anucleated. Then a cell had nucleus in the beginning. During the maturation process, gradually the nucleus has been removed. Such cell is known as enucleated cell. Of course, enucleated cell right now is a anucleated cell. One more time. From the beginning, there is no nucleus, anucleated. There is no nucleus at all, that is anucleated in general. Enucleated is a phenomena. By formation, it had a nucleus, but during the growth, that nucleus gradually disappear. Such kind of cells are known as enucleated cell. N means it was having, but right now it doesn't possess. This is called enucleated condition. Now, lack of nucleus will create lot of complications in the survival of sieve tubes. What is that? Simple. If a cell has no nucleus but living, the cytoplasm could not live forever. That's why we have seen one concept, RBC. It lives 120 days. After 120 days, it will go to the spleen, celebrates death. This is the life of RBC. Am I correct? But why did RBC die after 120 days? Because it can't survive more than that due to lack of nucleus. There may be several reasons are also there, but main reason, nucleus is absent. In that sense, come back to our sieve tubes. They are also do not possess nucleus, but they can survive for many years. If they are surviving for many years, the lack of the nucleus should be compensated by some other. That other is nothing but companion cells. Observe closely. There are some special parenchymatous cells associated all along the length of sieve tubes. These special parenchymatous cells are technically known as companion cells. They are companion to whom? They are companion to sieve tubes. In fact, these companion cells and the sieve tubes are siblings, which means they were developed from common mother cell. During the maturation, sieve tube had lost its nucleus, companion cell had retained. This nucleus of companion cell will be used by the sieve tube for its own requirements. How? Simple. This cell has enlarged nucleus and also the dense cytoplasm. These two cells will be having some sort of special mode of connections. These connections are known as plasmodesmata. Important. Plasmodesmata. Desmata. Now, I want here to recall one thing. In the earlier class, when I was talking about primary wall, secondary wall, I have explained one simple thing. There are minute gaps in secondary wall. Those gaps are called pit. There are gaps in primary wall even. That primary wall gaps, do you recall? What do we call the primary wall? The primary walls are known as pit fields. Am I correct? Pit fields, primary wall character, pits are secondary wall. You are saying companion cell is a parenchyma, and also sieve tube already mentioned it has primary wall. Both will have pit fields. The pit fields will help one cell cytoplasm get connected to another cell. In other words, the sieve tube cytoplasm and the companion cell cytoplasm get connected. That connection is developed due to plasmodesmata. By the help of the plasmodesmata, if any substance required for sieve tube to work, that substance would be generated by companion cells and given to sieve tubes. That's the reason why we often say companion cells help in maintain pressure gradient. Pressure gradient in sieve tubes. Pressure gradient in sieve tubes. Companion cell useful for maintain pressure gradient. See, we both are sisters. I don't have nucleus. I have become unnucleated. You have nucleus. Whenever I need, I will use your nucleus. Please say yes. This is the conversation between sieve tube and companion cell. So, companion cell provides all necessary components to sieve tube with the help of its nucleus. So, pressure gradient that it is necessary for the development of transportation of the substances within the sieve tube will come from companion cell. That's why companion cell sieve tubes always connected. Another thing, if we remove companion cells around the sieve tubes, gradually sieve tubes are going to die because they don't have their own individual survival. Okay? Next, after sieve tubes and companion cell, one thing we need to talk, that is phloem parenchyma. Phloem parenchyma. Whatever the structural details that you have noticed associated to parenchyma earlier, same detail, thin wall, living cells, and all those properties of parenchyma applicable here also. But in addition to that, one thing you should remember, phloem parenchyma absent in phloem parenchyma is absent in most of the monocots. Important, very, very important. Absent in most of the monocots. in most of the monocots, phloem parenchyma absent. They don't need actually. That's why they don't take any sort of parenchymatous tissue in their phloem. Who? Monocots. Why they say most of the monocots, not all? Reason, there is a member of the family called Graminaceae, grasses. In that family, most of the monocots do not carry phloem parenchyma. That's the reason why they say phloem parenchyma is absent in most of the monocots. Then, when you talk about parenchyma, typical functions, if you notice, storage. It store resins, latex, gums, etc. Sometimes they even store food, but already food is plentifully available in our phloem, so storing of the food may not be that much significant action. Keep it in mind. When we were talking about xylem parenchyma, we mentioned xylem parenchyma can store starch, oils, and secondary metabolites such as tannins. Tannins exclusive only for xylem. Tannins never ever will be stored by phloem parenchyma. Careful, gums are nothing but mucilage also. Sometimes mucilage are gums. Okay, that's about parenchyma. Then go to phloem fibers. Obviously, they are sclerenchymatous fibers. In that, fundamental characters will be same. What is the fundamental thing? Narrow cells, okay, because they are fibers. Next one, quite thick wall, very thick wall. The thick wall is lignified or non-lignified? Could you tell me? If I say there is a thick wall present in fibers, that thick wall of fibers is making the fiber to die. At the time, that thick wall is made up of what sort of material? Of course, lignified. Lignified. Done. After that, this is primary characters. Next one. If you are talking about special characters of phloem fibers, these are normal thing. Any fiber will have special features of phloem fibers. If you notice, they go like this. Their unbranched cells, very important. Unbranched cells, unbranched with pointed ends. Pointed ends. Cells are unbranched and they have pointed end. What is the speciality of the unbranched cells? Because of the cells are unbranched, they don't tangle easily. They don't tangle. They don't mix up easily. When you talk about such arrangement, very helpful for weaving the clothes, and also they are very good for textile industry. That's why these phloem fibers such as jute, flax, hemp, these are all okay, commercially useful. Commercially useful. That's about phloem fibers. But one thing you should never forget about phloem fibers, very, very important. The noticeable thing is phloem fibers absent in primary phloem. Primary phloem. Who is primary phloem? Who is secondary phloem? Anyhow, I will explain later. Keep it in mind. Phloem fibers are absent in primary phloem, but developed in secondary phloem. Those plants who can able to perform secondary growth, especially dicots, then only they can develop, then only they can develop They can develop secondary tissues. Whenever they develop secondary tissues, then only there is a possibility to have phloem fibers. This much information about phloem fibers. Fine. Then after the complex tissues discussion, let us talk about tissue systems. Tissue systems. What are the tissue systems and their story? Made up of more than one type of cells. More than one type of cells, which means varieties are there. It's not that just one group of cells can form tissue systems. Tissue systems are majorly formed for organs development. An organ, if you take for example root, just one variety of cell may not form a complete organ. They have to work together. That working ability will make them to present or stay together. That phenomena is known as tissue system. Now in the plant body, what are the tissue systems? If you want to classify, you need to go for criteria based on location where they are present, and also based on functions, what they do. Two criteria are there to classify tissue systems. One criteria is where they present, another criteria is what they do, their function. First one, based on the location and function, this is the only criteria. Keep it in the statement perfectly. Whenever they are asking what are the criteria are there to classify tissue systems, one is location, another one is function. Only two criteria are there. First one, epidermal tissue system. Epidermal tissue system. Next one, you will have ground tissue systems. Next one, vascular tissue system. Vascular tissue system. In that case, if I am talking about epidermal tissue system, what are the components present in epidermal tissue systems? If you question, I will answer like this. It is made up of epidermal cells, next one, and it has epidermal modifications. Epidermal modification, stomatal apparatus, next one, epidermal hair, These are the parts present in epidermal tissue system. In this, easy thing is epidermal cell discussion. Whenever you say epidermal cell, first thing, they are unilayer of cells, just one layer of cells. They arrange generally without intercellular spaces. No intercellular spaces. If you remember, when I was talking about parenchymatous cell, I have said there, generally parenchymatous cells do have intercellular spaces, but there is one variety of parenchymatous cell which arrange without intercellular spaces. That's called epidermis. Epidermis generally do not have intercellular spaces. Next, if you talk about epidermal cells of stem and leaves, they have a waxy coating on the top of that is called cuticle. Cuticle is a kind of waxy material. It prevents water transportation or water loss. Waxy substances are present. Is it okay? Now, presence of cuticle is a characteristic feature of stem, that to young stem and leaves. But if you talk about root epidermis, root epidermis. This is root epidermis, what I am writing. Cuticle absent. Cuticle absent. That's why the root epidermis is having a special name called epiblyma. What is that blyma? Epiblyma. Epiblyma. Blyma means covering. Epiblyma means outermost covering. The outermost covering of root, nothing but epidermis. But outermost covering of stem is cuticle. Generally, the layer of cells we say it as epidermis, but in account if you take, it is cuticle. Cuticle absent in case of the root epidermis, that way we will call that root epidermis as epiblyma. Clear? Then go back to epidermal modification. If I talk about stomatal apparatus, it's an epidermal modification. It consists of the following parts. One is stoma. Stoma. Next one is guard cells. Guard cells. And the next one is subsidiary cells. Subsidiary cells. Next, what are these parts? All these three parts collectively known as stomatal apparatus. Apparatus means equipment. What is this equipment and what is its story? The equipment here, it's a kind of operative mode of opening and closing of a special place called stoma. Stoma in Latin means pore. Observe how do you form the pore and how do you maintain regulation of that opening and closing of the pore. Observe. I will take some special type of epidermal cells here. These epidermal cells are called guard cells. Guard cells. This is a modified epidermal cell known as guard cell. Observe, this is a guard cell, another guard cell. Between the two guard cells, there is an existence of a small pore. That pore is known as stoma. Stoma singular, stomata plural. That's it. Stomata means many pores, stoma means single pore. How did you form this single pore? With the gap developed between two cells called guard cells. Let us analyze the guard cell story. Guard cells are specialized epidermal cells. They have uneven thick cell walls. One side of their wall, thick cellulosic depositions are noticed. Thick cellulosic depositions. Another side they have thin cellulosic deposition. This is the thin area. This portion you can call it as thick area. This is thick, that one is thin. Unevenly thick places. That thick places are around a pore. That pore is known as stoma. But interestingly, in case of dicot, these guard cells are bean shaped, and in case of monocots, these are dumbbell shaped. Dumbbell shape, that is guard cell shape. But whether you take bean shape or dumbbell shape, they will form a gap between two cells. That gap should be there, then only we will call it as stoma. If two cells are there, they are flat and joined, no gap. At the time, there is no development of stoma over there. So, in order to form stoma between the two guard cells, the guard cells either may be in the form of bean shape or kidney shape. They may be in the form of dumbbell shape. Dumbbell shape is a character to monocot God cells. Bean shape is character to dicot. That much simple. Next, you are saying they have a mechanism they can open and close the stoma. Listen, stoma is a specialized design for exchange of gases and also for the removal of water vapor. Be careful, I have seen many students construe that there is a exchange of water vapor. Water vapor never be exchanged. Only one thing will be doing with respect to water vapor. The water will go away from the plant to environment, but water vapor never come from plant to, environment to plant. Exchange never occur, but one thing will be exchanged, that is gases. Gases exchange could be possible, but evaporate to removal of water, water vapor will be happen by through stoma. Clear? Then observe the statement. When you talk about this stoma, sometimes it will close when they need not to evaporate to lose the water, and sometimes it will be open. In order to open and close the stomata, there will be a development of special arrangement in God cells. That arrangement is this. They have here and there the greenish plastids called chloroplast. Chloroplast. Why are you specially saying they have chloroplast? There is a reason behind that story. Generally, epidermal cells never perform photosynthesis. That is not their purpose. Their purpose is to cover up the plant organs. But God cells are specialized epidermal cell. They perform photosynthesis, or only epidermal cell perform photosynthesis. Photosynthesis. This is speciality. Okay. Now, why do they have to perform photosynthesis? Others could not do. That photosynthetic nature will make them sometimes develop some pressure. That pressure will allow the stomata to open. Sometimes pressure will be loose. Then stomata is going to be closed. Remember, increase the pressure inside the God cell is known as turgid. What do you call that? Turgid. If the God cell become turgid, turgid means excess quantity of a water in a one particular area. I have become turgid means I drink too much water, my stomach started to swell out. Then I will call it as turgid. Now, when the God cells become turgid, as a result of that, there will be a opening of stoma. Stoma opens. Then when the God cells loses water, that means they have got the water slowly go away. They will come to normal situation. That normal condition is known as flaccid. Flaccid. Stoma closes. Stoma get closes. That's the work of presence of chloroplast inside the God cell. But this opening and closing God cells cannot do by themselves. Around them or in the vicinity of the God cells, they have some specialized parenchymatous cells. These specialized parenchymatous cells are known as subsidiary cell. I said now, there is a requirement of water in the God cell in order to open and close the stomata. That water actually will come from subsidiary cells. So, in the vicinity means surrounding of the God cell, subsidiary cells are present. This subsidiary cell very much essential for supply of the required water for opening and closing of stomata. This is about how they can maintain a special apparatus that could help them to open, closes the stomata and also exchange of gases and evaporative removal of water in the name of transpiration. That's about that. Then go to epidermal hair. If you are talking about epidermal hair in the plant body, the epidermal hairs are of two types. One is trichomes. Trichomes. Who are trichomes? They are known as stem hairs. Stem hair. If a stem possesses hair, that's called hairy stem. If a stem has no hair, that's called glabrous stem. Not glamorous, glabrous. Glabrous means smooth. Observe, monocot stems generally do not have hair. Hair is something that could be seen only on the top of dicotyledonous stem. So, dicot stem, if hair develop, that is called stem hair or trichome. Their character, they are branched, sometimes branched, unbranched, unbranched. First of all, they also multicellular, sometimes soft, sometimes stiff, and even secretory sometimes. That means they can able to secrete variety of substances like scents and gums, etc. They can secrete. And main intention of trichomes to develop is reduce water loss by transpiration. If a plant performs excessive transpiration, it can wilt and eventually die. In order to avoid that problem, they have something that is called trichomes. Next, root hairs. Root hairs. I think you know one thing. When regions of roots, when we study, we have noticed one thing. Root hair are not present all over the root. Root hairs are exclusive for one particular area in the root. Do you remember what is that area? That is called region of maturation. Am I correct? Region of maturation. And other features are maturation zone. Other features, they are unicellular, always unbranched. And they don't have any other work. Only one work they have: increase surface area. Increase surface area of the root to absorb water and minerals. That's it. That's about root hairs. Like this, we have normal epidermal cell, modified epidermal cells such as stomatal apparatus, and epidermal appendages. Epidermal appendages means external outgrowth. External outgrowth of epidermis such as trichomes as well as root hair. Then go to next one, vascular tissue system. We have to discuss about the details of vascular tissue system. What do you mean by vascular tissue system? Xylem plus phloem together is known as vascular bundle. One bundle of vascular tissue you can identify, okay, xylem and phloem together. In that case, complex tissues, other name is called vascular bundles or vascular tissues. In that case, I am talking about arrangement of vascular bundles. Before that, first one, primary vascular bundle. Primary vascular bundles. Next one, secondary vascular bundles. Vascular bundle. Okay, who are primary? Newly formed. Okay, only for transportation. Their work is generally transportation. Okay, What about secondary tissues or secondary vascular bundle? Secondary xylem, here major part, which is called wood. Secondary phloem, commonly call it as bast. Now let me confirm these ideas. Okay, primary vascular bundle, secondary vascular bundle. Primary vascular bundles are universal. Whatever it may be the vascular plants you will study in the entire plant kingdom, all of them possess primary vascular bundle. But the secondary vascular bundle is quite rare. They will be seen only in those plants which perform secondary growth. Gymnosperms, dicots, where we will find this secondary vascular bundle. Now, hereafter, our discussion never goes to secondary vascular bundle. Our discussion is only primary vascular bundle because they are the one who are present everywhere and they are responsible for transportation mainly. Okay? Then primary vascular bundle, if you talk about, first I will discuss primary xylem. Whatever the discussions we will discuss about primary xylem, same things will also applicable to primary phloem. What I mean to say, listen. Xylem and phloem always synthesize together, work together, die together. Which means, if you see a place where phloem absent, xylem is ready to die. If there is a place where xylem is absent, there is no work for the phloem. In that case, xylem and phloem work together. That's why they are called a bundle. If I talk about there is a mechanism to mature xylem, the same maturation you could also see in phloem. Phloem, xylem, two times you have to discuss anyhow, but one time I will discuss maturation of xylem and formation of primary xylem. That would be sufficient to understand how phloem will also going to be formed. Primary xylem. Now, primary xylem, which is working in all plants to transport water and mineral, could be divided into two categories: protoxylem, primary xylem itself, protoxylem. Next one, metaxylem. Who are this proto and meta? New or newly formed, just only formed. These are non-functional. They can't conduct water. Non-functional because narrow lumens. Lumens are in the vessels very narrow. Since very narrow lumen, they can't conduct water. Who? Primary xylem. Protoxylem. Protoxylem, metaxylem is a kind of primary xylem. Now, metaxylem, I am counting. Metaxylem, matured one. Matured one. Matured for what? To transport water. Next one. It is functional. It is the one which can able to transport water and mineral. And also they have wider lumen. Lumens are very wider. Where? Metaxylem. Lumens are very narrower. Where? Protoxylem. Keep it in mind. Protoxylem just formed. Metaxylem already formed, working. Both are belongs to primary xylem. Primary xylem everywhere. Secondary xylem rare. Primary xylem just formed from cambium, protoxylem, matured and conducting water called metaxylem. There I am talking about xylem and protoxylem, metaxylem appearance. Their appearance would be changes with respect to type of organs. For example, if I choose stem, in the stem, I am taking cross section of a stem. This is a cross section. That means I will take a stem, cut it transversely and watching from the top. You take the top view of a stem then you will find metaxylem, which is somewhat larger, present towards the periphery. Protoxylem, which has narrower lumen, present towards center. Observe this. In this arrangement, you have to remember this is metaxylem and this is protoxylem. Protoxylem has narrower lumen. Metaxylem will have wider lumen. Who can conduct water? Majorly metaxylem will conduct water. Both are protoxylem only. In that case, if protoxylem present in the center, metaxylem present in the periphery, this mode of arrangement is known as endark protoxylem. Endark protoxylem. Protoxylem present inside the center or inner side of the stem. This is the characteristic feature of stem. If somebody given a piece of plant tissue to you and they are asking you to identify from where this plant tissue has been collected, directly you have to see what is the location of their protoxylem. If the protoxylem present in the center, that should be stem. If the protoxylem present outer side, which means I am talking with respect to root now. In the root arrangement, their arrangement, if you see, arrangement goes like this. Protoxylem, the young one, present towards the periphery. Metaxylem, the old one, present towards center. Observe opposite direction. This is proto, and this one is meta. Metaxylem center, protoxylem periphery. This arrangement is known as exarch protoxylem. Exarch protoxylem is the characteristic feature of root. Here, root and stem significantly varies their mode of maturation of xylem. What is that? If you take stem, maturation will start from center and go periphery. But if you talk about root, maturation will be started from the periphery and it go towards center. The maturation of the xylem, this is center to periphery, centrifugal, and the peripheral to center. The maturation is centripetal. Okay, that's about arrangement of proto and metaxylem. Is it clear? Whenever you talk about this arrangement, that arrangement signifies, okay, there is a difference in development of xylem in stem and root. Stem has one mode of development, root will have another mode of development. Next, you are saying xylem maturation. What about phloem maturation? The interesting, if the xylem is having endark protoxylem, the same rule that phloem has followed. Xylem is endark, phloem is exarch, not possible. Wherever you have seen there is a protoxylem present in the center, that arrangement will also signifies protophlloem is also present in center. Opposite direction not possible because they grow mature in the same direction. Think about that. One side metaxylem is present, which is ready to conduct water. Parallel to that, you have kept protophlloem, which is not conducting. Both will fight and the plant will die. That's why always the orientational maturation will be same for xylem and phloem. Next, go for another mode. Types of vascular bundles. Types of vascular bundles. This is based on maturation, based on arrangement, based on arrangement. It follows different direction like this. I am taking a stem. For example, dicotyledonous stem I am taking. In the stem, matured one. This is proto. I mean, here I have discussed about how they will mature. Now I am talking after maturation, what is the arrangement? Types of vascular bundles based on arrangement of xylem and phloem. In stem, cross section of the stem I have taken, that is transverse section, watching from the top by transverse section. Over there, I could see there are vascular bundles like this. Xylem and phloem are arranged like this. In this arrangement, there is a xylem, there is xylem, 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 and also phloem. Observe phloem. This is phloem. These are vascular bundles. Xylem and phloem together I am noticing how they are arranged, not matured, how they are arranged. Now, if you talk about xylem phloem arrangement, observe here closely. This entire area belongs to phloem. The central area belongs to xylem. Observe, I am going to draw a radial line. Radius I have drawn. If I draw a radius, that radius can penetrate xylem and phloem together. Such kind of arrangement of vascular bundles are called conjoint vascular bundles. Conjoint vascular bundle. That means xylem, phloem arranged on same radius. In the stem, if I am going from one side, first I will encounter phloem and I am seeing xylem later. Or else, if I come from the center to periphery, first I will encounter xylem. That means xylem, center, phloem, periphery. Phloem, peripheral. This arrangement is known as conjoint vascular bundle. Wherever you see a stem, whether it is a monocot, dicot, whatever it may be the cot, they will have in the stem conjoint vascular bundle. But if you analyze further about this conjoint vascular bundle, sometimes what happen with respect to conjoint vascular bundle, I am talking conjoint vascular bundle. Some conjoint vascular bundle where there is a xylem and phloem and the phloem. Xylem and phloem present. So xylem center, phloem periphery. Okay, that's one arrangement. Observe, there is another mode of arrangement we could see. That arrangement says this. There is a vascular bundle in this. Xylem present, center, xylem, phloem present, periphery, phloem present, periphery, xylem, phloem present, but they are present in the same radius with some sort of special type of cambial cells present between them. In this case, this is called conjoint collateral closed. If intrafascicular cambium When I was talking about meristems, I have given several places this intravascular cambium. Now, at the time, I have also said intravascular cambium is a kind of primary meristem. This is present in primary tissue between phloem, xylem. Okay, phloem, xylem. That's called, it's a kind of primary meristem. By the presence of intravascular cambium, this vascular bundle can grow further. Such kind of vascular bundles are called open. They are open for what? Open to grow. They can grow further. Which one? Conjoint, collateral, or conjoint vascular bundle, which are having intravascular cambium between them, is a kind of open type. Open type means they are capable to grow in future. If you stimulate them, they can increase the vascular bundle size. That's why I say they are open to grow. Open vascular bundle. You make this one, whatever you do, it cannot grow. Grown over. This is called closed. Closed vascular bundles present in monocot stem. Next, open vascular bundles are present in dicot stem. Of course, conjoint vascular bundle is a characteristic feature of stem. In monocot stem, they are closed. They can't grow because lack of intravascular cambium. In dicot, they can grow. What is the reason behind their growth? They possess a special type of vascular cambium called intravascular cambium. If you closely observe, I have given one more name for it. That is fascicular vascular cambium. Fascicule means bundle. Okay? Fascicular vascular cambium, or simply you can call it as intravascular cambium. It could be seen in dicot. This could be seen in monocot. That's why there is a variation in arrangement of primary xylem and phloem with respect to stem and root. Let us see root. What kind of arrangement? In the root, if you are observing the arrangement of xylem and phloem, you will get one arrangement like this. This is root. Okay, in the root there is a presence of xylem, but this xylem protoxylem towards the center, metaxylem towards periphery. If so, observe, this is xylem. Only xylem not sufficient, right? There will be also presence of phloem. This is phloem tissue. Here you can see that when you have xylem and phloem, they are not connected. They are not at all connected. Xylem is in one radius. If I draw radius on this, xylem is in one radius, phloem will be on another radius. Xylem and phloem are not found in same radius. This kind of vascular bundles are characteristic to root. They are called radial vascular bundles. Radial vascular bundles. You can say that xylem, phloem are present on two different radius, but between them empty space doesn't exist. What is present? Between them there is a presence of special type of parenchymatous cells, that's called conjunctive tissue. Conjunctive tissue. This is about the root arrangement of vascular bundle. This is about stem. Now, this is about how vascular bundles are arranged. Next, finally, we need to talk about ground tissue systems. If anybody asks you what is ground tissue system, there you will get except epidermis, and vascular bundles, except epidermis and vascular bundles. Remaining all tissues are denoted as ground tissue system. In that, what you get? Hypodermis, cortex, endodermis, pericycle, pith, medullary race, etc. All of them are having ground tissue organization. In that, very importantly, one thing we have to talk, that is, leaf mesophyll cells. The leaf mesophyll cells, who are well known for photosynthesis, leaf mesophyll cell, they are responsible for photosynthesis. They are a variety of ground tissue. Next one. When you talk about ground tissue, what are the tissues you can count there? The tissues, majorly parenchyma. The tissues over there, majorly parenchyma. Rarely sclerenchyma. Very much rarely you will find collenchyma. Apart from that, okay, you don't find extraordinary organization in ground tissue system. Okay, this is about ground tissue. Next, we have to talk about the TS of dicot root and monocot root. Transverse section of dicot root. In that, the example of dicot root transverse section is sunflower. Generally they take sunflower. These are easy for us to analyze hereafter. Easily we know what are the tissues, where they arrange, how they arrange, all of them to be collected in one place, and you have to study organs. Learning about tissues and their characterization might be difficult, but you can arrange them very easily in different places to make them organs. Now I am forming dicot root. First, outermost layer is obviously epidermis. What are the characters of epidermal cells? They do not have intracellular spaces. Now, since you are talking about root, do they have hair? Yes, they have hair. They are root hairs. The epidermis, does it covered by cuticle? No. Since it does not covered by cuticle, you can call it as epiblyma. What blyma? Epiblyma. That means it is a layer which should be noticed as outermost layer of a root. Okay, only root epidermis is known as epiblyma. Other than that, no. Next one. Below that epidermis, you can see general cortex. These are large parenchymatous cells with arranged intracellular spaces, etc., etc., etc. They are cortex. Okay. Innermost layer of the cortex has a special significance here. The innermost layer of cortex. You know the innermost layer of the cortex, what we call? Endodermis. The innermost layer of cortex. Layer of what, ma? Cortex. Okay. This innermost layer of the cortex, known as endodermis, it is also made up of parenchymatous cells, but the parenchymatous cells are barrel shaped. They arrange without any intracellular spaces. Next, in the endodermis, we have some special kind of appearance. That appearance Their radial walls. What do you mean by radial wall? This is a radial wall, radius, and this is tangential wall. Both on the radial walls as well as on the tangential walls, they have some specialized water impervious coating. That water impervious coating is known as Casparian strip. Do you remember that word? Casparian strips. Casparian strips will prevent water loss from inner cylinder to outer area of the root. This area is known as Casparian strip. Casparian strip made up of what? Suberin. Suberin is water permeable or impermeable? It is water impervious means not allowed to pass. Next one. Endodermis, inside there is a single or multiple layers of generally unilayer. Okay, sometimes multiple layers also would be there. They have a special kind of layer of thick-walled parenchymatous cells are present. Thick-walled parenchymatous cells, that is known as pericycle. Which cycle? Pericycle. Cycle here, vascular cycle. Around it, there is a presence of a thick-walled parenchymatous cell. The thick-walled parenchymatous cells are known as pericycle. Then what is the significance of the pericycle? Pericycle is very important. It is responsible for formation of lateral roots. Responsible for lateral roots formation. This is very touchable thing in that anatomical features. What is that? When you talk about lateral roots, which are nothing but branches of root. Have you seen branches even in stem? Yes, we have seen branches in stem. Could you recall from where we have got that branches? A stem produced branch. Am I correct? The branches are developed from where? The branches of stem are developed from axillary bud. When I was talking about axillary bud, I said this. Some amount of shoot apical meristem left over at the axil of leaf that behaves like axillary bud. That axillary bud in future, it can form a branch or it can form a flower. Now, branch is developed from axillary bud. That axillary bud is external or internal organ? It is external. One thing you remember, bud, some kind of structure that could be belongs to stem. On the root, you will never find a bud. In fact, bud is nothing but a concised stem. Stem concises and form a bud. So, bud, external body which produces a branch. Now, a root want to produce a branch. That branch never developed from bud. It is developed from pericycle. That is an inner area of our cortex. Even to the cortex, it is very inner place. That's why branches of root, endogenous in origin. Endogenous in origin. Branches of root are always endogenous in origin. But branches of stem, exogenous. Root hair, exogenous. Stem hair, trichomes, exogenous. But root produces branch. Very important. Root produces branch. That branch is always internal. Stem branch, external. Then go to inside. Here one thing, one word you have, all the tissues inside endodermis. All tissues inside endodermis has a name called stele. Not that steel. Here steel means cylinder or central cylinder, not that steel, steel, not iron. Steel means center. Observe the story. When I go to inside the steel, I will first encounter pericycle. After that, I will find xylem and phloem. In the root, there is a presence of exarch protoxylem. Am I correct? Root exarch protoxylem. Stem is endarch protoxylem. Remember. Next one. They do have phloem. Phloem also same story. Exarch protophlloem. Next, this is phloem. This one is xylem. Okay. The center part, what we call this area, it is known as pith or central area. And this portion, do you remember? Xylem and phloem are separated by conjunctive cells. This is about anatomical features of dicotyledonous root. But what about monocotyledonous root? The features of monocotyledonous root, dicotyledonous roots, almost same with some slight variations. Now I am talking about the variations. Simple. Dicot root. First, in the dicot root, if you notice, 2 to 4 xylem bundles. 2 to 4 xylem bundles. 2 to 4, that's it, not more than that. 2 to 4 xylem bundles only. Okay. Next one, they have inconspicuous pith. Okay. Pith is very small and it is inconspicuous. Next one, they do have, observe here, conjunctive cells. Later there will be a development of vascular cambium for the secondary growth. So secondary growth can occur. Next, monocot root, if you check, there you will have more than 6 xylem bundles. This arrangement is called as polyarch condition. Next one, conspicuous pith. Next one, secondary growth absent. Remaining all features of monocot root, dicot root will be same. Okay. This is about dicot root and monocot root. On Sunday, I think you will have exam based on up to here. Okay. Any doubts? If you have, I can able to explain. Come on, paste it there. Any doubts? Come on. Are you able to follow me? Am I going faster or I am going somewhere, something? You just give your feedback because we are in live class. Anything you need? Come on. Everything going well? Or do we need any adjustments? Your suggestions are acceptable. Come on. Okay. Thank you. We will meet in the next class. Do well. Okay.